Today we're talking about chemistry of life, proteins. This is taken from your Nelson biology textbook, page 39 to 44. Behind the slide here, what you're looking at is a protein called kinesin. And kinesin is absolutely necessary for your cells to survive. What it does is it is a carrier, and it looks like it's walking right now. This is actually what it looks like, and it carries vesicles. You can see the cell membrane kind of moving as it tugs it along. And this is made out of amino acids, and it's using energy to move things around your cells. Proteins are necessary for just about everything inside your body. These things include enzymes, which are your cell's machines. They do various jobs. Again, enzymes are absolutely necessary for life. They do various things in your body. Immunoglobulins, and right now my body has immunoglobulins, and I've tried to fight off COVID. And they're doing a good job, hopefully. We have hemoglobin, which carry oxygen around your blood. And you got keratin, which is what your hair is made up, and fibrin, which is in your blood, which helps your blood clot. That's just to name a few. There is literally millions, hundreds of thousands of proteins out there in your human body. It's really incredible. So proteins are made up of amino acids. And then this diagram right here, I'm showing you the primary structure. All these little letters there, each one represents an amino acid. And what's happening is when we arrange them and link them together, we get a polypeptide chain. And then that polypeptide chain is going to fold. And when we fold, we mean bend. And this is the secondary function. So eventually what your protein will have is a very specific shape, and that's going to lead to a very specific function. If you're going to learn one important thing from this lesson, it would be specific shape, specific function. So proteins can have an indefinite number of different shapes, so therefore they can have an indefinite numbers of different functions. So don't write this down yet, but let me give you a rundown of how proteins get their specific shape, which enables their specific function. So the primary shape that it gets from is just a sequence of amino acids bonded together. We call this the primary protein structure, okay? What will happen next is there's going to be some H bonding in there. There's going to be some interactions there that are going to cause it to form different three-dimensional shapes, like a helix shape, okay? Or what we call a beta pleated sheet shape, which is just basically, it's like a sheet of paper, but it's bent kind of at diagonals, okay? Then what you're going to have is you're going to have a tertiary protein structure, which is going to be, there's these side groups on the amino acids, they interact, they're hydrophobic or hydrophilic, and they'll want to come together and they'll make this three-dimensional shape, okay? At this point, it's like a protein globule. And if you take a couple different of those and you put them together, you get what we call the quaternary final shape, which is the actual active protein that has the specific function because of the specific shape that it has. So what are some of those specific protein functions? One of them is structural support, like the cytoskeleton. This is an image of some cells. It's a microscope image. And I believe it's dyed so you can see it. All the green lines that you see in these cells, that is the cytoskeleton. It holds it together. It's like sort of like the connective tissue within the cell. Okay, you also have connective tissue in your body, like your bones and your ears, and you have protein that makes up those, and it gives you that structural support. You may also remember that cell membranes do not allow charged substances to go through them because they're nonpolar. Well, you need special protein channels that allow ions to pass through, or big molecules, and they're made of protein. Also, obviously you've heard of diabetes before, and that relates to insulin. And this is actually an image of insulin. All these ribbons are different alpha helixes and beta plated sheets, and it's a very specific shape, and that is a cell signaling molecule. It allows glucose into your cells, okay? And that is a protein. We talked about kinesin already, and that involves cell movement, moves things around. And there's also these proteins can also change their shape, which is really important. Right here we have an image of hemoglobin, which is the primary protein of your blood cells. And what happens is it will actually change its shape to fit oxygen or carbon dioxide, depending on what it's doing at the current time. And that change of shape allows it to do its function a little bit better. It allows it to capture more oxygen, for example. And that shape change is really important. And because of that, because proteins can change shape, they can also become enzymes. And enzymes are little molecules of protein that will change their shape to do a chemical reaction. They have these little active sites which allow reactants chemicals to fit in them, and then the enzyme will change shape and will make those chemicals react. So a lot of different structures mean a lot of different functions for these proteins. So different amino acids will link up to form this primary structure of a protein. And so they link together. So what is an amino acid? What are each one of these things? Well, they're made up of an amino group, which is the NH3 group, or sometimes it's shown as NH2, and the carboxyl group, which is the acid group, which is the COOH. So you can see that the H has been transferred from one to the other, and that's because it happens at a pH of 7. Generally, I like showing the amino part as NH2 and the carboxyl group as COOH, but that's okay. You can draw it either way. Now, when this middle carbon is the most important thing. All amino acids have an amino, they have a carboxyl group, but they have a different R group. And there's actually 20 different R groups that are possible in your body. And you put a different R group on there, you're going to change the properties of that individual amino acid. You can add an R group that's hydrophilic, so it's polar, or you can make it hydrophobic by throwing something that's nonpolar on there. You can make it acidic by putting a carboxyl group on the actual R group, or you could make it basic by putting an amino group. So again, an acid group is a carboxyl group, and a basic group is an NH2. And you can put those on the R as well, and that's going to change the properties of that amino acid. So there are 20 amino acids that are most of the animal kingdom. There's actually 22, I think, in the world. The majority of animals will use just these 20 amino acids. And you need a certain amount in your diet. So the essential amino acids, there's 9 essential amino acids that you need to consume in your diet for you to be able to make your proteins. The rest of them, the other 11, your body can manufacture on its own. So let's see if we can draw an amino acid, make sure that we know how to do that. So the way we draw an amino acid is we just do our NH2, okay? So keeping in mind, we got a lone pair on that N, okay? We got a carbon, we've got our R group, right? And then we have our acid group. So we have to be able to know how to draw these guys. You don't have to show the lone pair. Sometimes you can just show it as OH there, and we recognize that as an acid group. Now, if we change that R group, we change its properties. So you don't have to memorize any of these. You'll be given a sheet of this sheet, so you can look at it. But there's plenty of nonpolar amino acids. You can see the R groups all consist of just basically hydrocarbons, and they have nonpolar bonds, and so therefore they're nonpolar side groups. There's some, like we're incorporating sulfur here, methionine, phenylalanine. This is a carbon here. So if you don't recognize this, this is known as a benzene ring in organic chemistry. But those are all carbons, and there's hydrogens attached to it. So anytime you see those lines, it's just carbons and hydrogens, okay? Tryptophan, proline, they're all nonpolar. We have polar amino acids, which are going to have, you'll notice they'll have an OH functional group on there. Like threonine has an OH on it, cysteine has a sulfur and S. That is an electronegative atom, and that is going to create dipoles, and that's going to make it polar. You have an NH2, an amide functional group. That's what we call an amide functional group there, and that is also making it polar. You can make them acidic by adding a acid functional group. Sometimes we show an H on there, so COOH. You can make it basic by adding an amino functional group on it. So aspartic acid, glutamic acid are acids, obviously, and lysine, arginine, histidine are basic because of the N's, the NH, the nitrogens that are in its structure. So to create this primary structure, we have to link together amino acids. And to do that, we take the carboxyl group of one amino acid, and we combine it with the amino group of another amino acid, a different one, And you're going to, anytime we do anabolic synthesis, it's dehydration synthesis, we're removing water. So in blue here, you can see that we're removing water. And when we do that, we create a new link and an amide linkage or a peptide bond that exists between these things. Now, when you collect these guys together, you repeat this process over and over again because every amino acid has an amino and a carboxyl end. You end up getting a long chain with lots of amide bonds. So if we look at this, we have 1, 2, 3, 4 amino acids. This GLY is referring to the side group for a certain amino acid. Okay, so those are specific amino acids. We have an amino end where it's not bonded to anything, and we have a carboxyl end. Sometimes we call it a amino terminus. Terminus means end. So eventually what will happen is you'll get these chains and the next structures, they're going to fold together. But one thing that you need to be able to do is actually to be able to draw an amino acid forming this peptide bond. So a question would be like, okay, I'm going to pick 2 amino acids. Let's pick simple ones. Let's pick glycine, which is the simplest amino acid. It's just got an R group. And let's pick serine and let's bond them together. So I'm going to draw them out and just on the diagram here, and then we're going to show what it looks like when they combine together. Okay, so I've highlighted the blue here, and then I've highlighted as the amino groups, and I've highlighted the carboxyl groups for you to see, and then the R group is highlighted in yellow there. So what will happen is you get a dehydration synthesis for this to occur. So you're going to remove water. That's what dehydration means. And you look at your 2 functional groups, the acid group, and you look at the amino group, and you're going to remove that water. And when you remove that water, there is a new connection, and you're going to make an amide linkage. So I'll start with the amide linkage, which is a double bonded O. That's this right here. And then I'm going to have an N, okay, which is this right here. I'm going to draw its H off the top, which I just drew there. I'm now going to complete the rest of this molecule here and add it onto that N. So it's going to be C, double bonded O, OH, and then off of that, sorry, wait, I made a mistake there. There's 2 carbons there. Let's take a look. So there's 2 carbons, C double bonded O, OH. And then off that center carbon, we have a CH2 and an OH here. So that's this part at the bottom. So I've just repeated that. Reminder again, there is an H off that center because it has to make 4 bonds. So I'll show that there for you so you feel better about it. Then what I'm going to do is I'm going to continue to draw this part of the molecule on the other side. Okay, so we're starting at the C double bonded O, which is right here. And now I got a C, an H, and then I go to NH2. And you can just draw it as NH2 at this point. You don't have to show the H's splitting out there. So this is the amino terminus, and this is the carboxyl terminus. Terminus means end. And again, you have different functional groups sitting out there. That's the H from glycine, and this is the CH2OH from the serine. And that's it for that. Okay, let's run through the way that this protein folds one more time. So primary structure is the amino acid chain. Secondary structure is going to be the different shapes, either beta pleated or alpha helix. Okay, so these shapes form because of H bonds. Then you have a very specific structure which results from the R groups. So the R groups that we're going to learn are going to be either hydrophobic or hydrophilic, and they're going to cause that, not just these beta pleated sheets and these alpha helixes, but they're going to cause that chain to fold up so that the hydrophobic stays in the middle and the hydrophilic stays on the outside. And that's just going to be one protein subunit or a globular protein. And then finally we combine that with another globular protein and we get the quaternary structure, which is more than one polypeptide subunit or more than one globular protein. Okay, so summarize again, primary structure, no function. It's just a sequence of amino acids, 20 different amino acids. So there's a different amino acid possible at each one of these spots on the chain. So there's a wide variety of possibilities. And this is really going to determine how it folds eventually, and all that other stuff because of the side groups that are on each one of those individual amino acids. The secondary structure results from the hydrogen bonding that occurs within the polypeptide chain itself. So the polypeptide chain is going to fold in on itself in an alpha helix, or it's going to turn into one of these beta pleated sheets, okay? So the tertiary structure, you now start having a function. And the tertiary structure forms from hydrophobic and hydrophilic side groups. Hydrophilic groups want to be on the outside, hydrophobic groups want to be on the inside. You also get additional hydrogen bonding, which kind of supports this three-dimensional shape. You can get ionic bonding. Certain groups, like the acids, will have a negative charge, and depending what the pH is, the bases will have a positive charge, and they can form an ionic bond, supporting that three-dimensional structure. An even stronger bond is something called a disulfide bridge, which happens when you have a lot of cysteine. So if your hair has a lot of cysteine in it, this is the amino acid with this sulfur, this SH, two SHs can come together and they'll form what we call a disulfide bridge, which is right here. Okay, so two side groups are getting rid of their H's and they form this disulfide bridge, which really supports the structure of the tertiary structure, okay? And when you straighten your hair, what you're really doing is you're actually breaking disulfide bridges and you're reforming them. If you have curly hair, it means you have a lot of cysteine in your hair, in the protein structure of your hair, and that's causing your hair to have a lot of disulfide bridges and making it curly. Finally, the quaternary structure is when you have two different polypeptide chains that are folded to a tertiary structure, and you got these two shapes, and you're coming together to form this quaternary shape. So there's different subunits. We call them subunits when they're from the same polypeptide chain. And something like hemoglobin, I believe it has 4 different subunits that make up hemoglobin, and they all come together and perform a specific function. So a good analogy for this is that the primary structure is like the alphabet, okay? The secondary structure are like the words, and then the tertiary structure are like the sentences, and then the quaternary structure is like the paragraph. You're getting more specific function and clarity in your speech when you go from words, alphabet, to paragraph. And similarly, you're getting more specific function when you go from primary, second, tertiary to quaternary. That, thanks to Heather Laurie for that one. That's a different bio teacher's analogy, and I stole that from her, and that's awesome. So I look forward to seeing you guys tomorrow. If you have any questions, please feel free to fire me a message on Edsby. Thanks. Bye.